about systems genetics. So this is the last lecture in the module about genetics. So we're taking it to the systems level. And we're also going to be talking about electronic health records. So we're going to introduce linear mixed models, talk about heritability definitions, dive into LD score regression, and then electronic health records and GWAS integration. And we're very fortunate to have two guest lectures by Dr. Alkis Price over at HSPH and Dr. Manuel Rivas over at Stanford. So we're going to review very briefly what have we seen so far in this module, and then dive into linear mixed models, polygenic risk scores, heritability, and then talking a little bit about omnigenic models, and then turning to LD score regression. I have some additional slides on advanced topics that we obviously will not have time to cover, and then we'll switch over to Alkis and Manuel. So just to review briefly where we stand, we've talked about common and rare variants, strong effects and weak effects, and polygenic risk scores, the application domains of GWAS, and linkage analysis. We've talked about linkage disequilibrium, which is your friend if you're trying to just simply find where are the blocks of association, because you can genotype only a subset of SNPs and then find the blocks, but it becomes your enemy when you're trying to actually find map what are the causal variants that are driving these associations. And that has been very much the challenge of understanding the mechanism underlying genome-wide association studies, and why it's so difficult to interpret them, because the vast majority of these associations are non-coding, and they have multiple SNPs, meaning that we don't know the causal variant, we don't know the target gene, the cell type of action, the relevant pathways, and so on and so forth. So what we've been talking about in this module is how do we integrate epigenomic information for fine mapping variants, and then RNA information, variation information, and deep learning models for sequence function to start predicting driver genes, regions, and cell types to actually get the circuitry underlying these disease loci. And we saw how these apply to specific domains to actually allow us to both uncover the circuitry and then reverse the circuitry. And then last time we looked at how we can actually use GWAS enrichments at the genome-wide level to find relevant tissues using just a very simple enrichment analysis. Today, the guest lecture by Dr. Alkis Price will dive into one of the most prevalent methods for doing that, namely LD score regression. We talked about how we can use these enrichments for prioritizing the posterior probability of genetic variants, given the many genetic variants in association, how we can use epigenomic information for prioritizing these variants, and how we can use this information to also prioritize subthreshold new disease loci by training on the genome-wide significant loci, and then applying this to subthreshold loci. And then we saw how we can use vastly increased numbers of tissues and cell types to start dissecting the components that contribute to complex traits like coronary artery disease into their liver-specific, heart-specific, and pleiotropic components. And then we turn to EQTLs and how we can trace the intermediate effects of these genetic variants down the path of causality, and how we can start using imputation for predicting correlations between the genetic component of these intermediate molecular phenotypes and ultimately disease. And then we saw the nuts and bolts of EQTLs, and specifically how we can look at expression as a linear regression prediction from genotype and covariates. And we saw this EQTL model that doesn't just use these regression coefficients, these effect size coefficients beta, as products of the genotype of that individual, namely 0, 1, 2 risk alleles, for example, or alternate alleles, but also utilizing these covariates. Today, we're going to be expanding that to add additional population stratification effects. And in particular, we're going to be looking at linear mixed models for both GWAS and for EQTL calling. So what are linear mixed models? So we're basically going to be looking at both the fixed effects and the random effects that are resulting in the phenotype of interest. So the setup is very simple. We basically are trying to predict the set of phenotypes for, let's say, 1,000 individuals using their genotypes for those 1,000 individuals across millions of locations across the genome. And these theta vectors are basically telling us what is the contribution of every one of these genetic variants, plus some error. Okay, so this is the basic framework within which we're going to be looking at prediction of phenotype. That phenotype could be expression or it could be height, using some fixed component and using these genotype dependent components. So who's with me here on the basic foundation of predicting the phenotype of a person based on the number of alternate alleles that they have and the effect size of each of these alternate alleles across all SNPs in the genome and across all individuals in our cohort. Lovely. So we're at 53, 41, 600. So this is exactly the same thing written in linear algebra form. So we're basically going to be applying the same scaling factors, theta, on every single genotype, and then adding up all of the genome-wide effects for each person. Now, the question is, how is the noise distributed? And we can basically think of the noise as simply distributed across identically distributed individuals, where every single one of them has a noise centered at 0 with some, squared covariance matrix, which is basically a diagonal matrix that is simply scaling by a set of scalars, which are the variances for every individual, the same identity matrix. So all of these are IID, basically. The problem is that this actually might not be true. So what we want to actually think about is, in addition to this noise vector that is completely independent across individuals, we want to account for random effects that might be captured by a kinship covariance. What is a kinship covariance? That basically tells us how much of the genetic variance that we've measured is shared by every pair of individuals. So this is simply a vector that basically tells us for every individual how much sharing do they have in their genetic loci. So we basically have the IID component, and we also have kinship component. And for many, many genome-wide association study problems, the most influential random effects stem from population structure. So what we're going to be doing is modeling those as a global effect stemming from that covariance kinship matrix. So we're going to be using a Bayesian approach to account for these random effects by saying that they are distributed according to the kinship matrix, and using this Bayesian approach that addresses and removes the uncertainty by averaging it out. So we're going to be integrating over all of these unknowns to, using this kinship derived noise to basically say what is the probability of each of the phenotypic effects being driven by that covariance matrix. And that's where the linear mixed model will come in. So basically, we'll be able to look at the phenotypic consequences as a linear function of the genotypic matrix plus an error component that incorporates both the IID error that we saw before and the kinship components, which is the relatedness. Hey, Alkis, you're here already. So, Alkis, what time is your guest lecture starting? I thought it was 1:30, but I'm vaguely remembering that
I thought you had discussed switching to 1:15. Either way is fine with me. I do have a hard stop at 2 for my course.
Okay, so I will try to wrap up quite rapidly because I think you're going to be covering a lot of these things. So I'll try to wrap up in the next few minutes. So the basic foundation of these linear mixed models is that you're going to be building a joint model of all the SNPs and how do they explain the total heritability of a particular trait according to this infinitesimal assumption, which we can estimate using this restricted maximum likelihood model. And that avoids using an ML fit, an estimate for each of the parameters, and instead uses transform data so that all of the nuisance parameters have no effect. And we're going to be using this variance components analysis, this random effects model that allows us to capture these transformations. So the beauty of all this is that it actually works despite not knowing the actual causal variance. So and we're going to talk about heritability very shortly. So what this is allowing us to do is actually start capturing a lot of additional variation. So you can use this in two different ways. You can use this by predicting the effect on each of these, for example, intermediate molecular phenotypes from the instantiated SNPs of every individual. And that's very similar from the way that we talked about last time. Or you can directly use the linear relationships between the SNPs and the expression, and the linear relationships between the expression and the trait, to actually not have to have individual genotype variables instantiated for every one person, but instead combine these effects through the LD relatedness of all your individuals, and through the effects of each of your SNPs and each of your genes on the ultimate phenotype. So you can use this to start predicting what are the effects of every SNP on the expression level, for example, for these intermediate phenotypes, using EQTL-based SNP effect, and then using observed factors, which are your covariates that you have observed in your cohort, but also hidden factors that can be learned de novo from your cohort. So you can use these models with an uninformative prior, or you can basically say, well, we expect these parameters to be centered at our maximum likelihood estimate or some other estimate, and then we have some potential noise surrounding that, and you can use prior distributions that match this with either a smooth or a very sharp centering on those, and then search through this parameter space to start inferring the parameters that you would like. And in fact, one of those strongest effects that is acting at the global level is in fact stemming directly from the population differences. So this has been shown in fact by Alkis, who's here, to be one of the strongest effects driving these genetic matrices across, for example, the European continent, where if you look at the principal component analysis of the genotype matrix in absence of any kind of phenotypic effect, you basically see that the position of all of your samples according to the first two principal components of variation is in fact recapitulating the geographic location from which they came within the European continent, indicating that there are these global cross-cutting effects on your genotypes. So we can use all of this to now correct for those matrices and then start inferring that. So I'm very tempted to switch directly to Alkis, and maybe we can cover this at the beginning of the lecture next time. Maybe one of the key points that I want to capture, I think that will be very helpful in preparing for Alkis's presentation, is the concept of heritability. The concept that we can basically ask how much of the phenotypic variance can we explain using additive effects, using dominance effects, and using interaction effects. And a lot of GWAS is underlying, is making this underlying implicit assumption of additiveness, whereby if I keep adding independent effects of every SNP in the genome, I will eventually capture some fraction of the total heritability. And this heritability is in fact allowing you to start asking how related are any two individuals phenotypically based on how related are any two individuals at the genotype level. And this actually gives you a very powerful approach for computing heritability as simply the ratio of phenotypic variance on genotype, of phenotype similarity on genotype similarity. That's where it gets actually very interesting, because you can actually start asking, if I partition my genetic relatedness into subsets of the genome, instead of asking about this relatedness in the entire genome, I could ask for relatedness in a subset of the genome. For example, only chromosome 1, or only chromosome 2, or only chromosome 3, chromosome 4 only, and so on and so forth. And what was really remarkable is that we found, or at least the field found, that the longer the chromosomes, and here they're ordered by their length, they're numbered by their length in the human genome, the longer the chromosomes, the more variance they explain for many, many of these complex traits, suggesting that phenotypic, the phenotypic basis of disease is almost uniformly distributed across the genome. And you can basically take different 1 megabase genome chunks and then see how much heritability is captured there. And that is actually at the foundation of partitioning the genome in different ways. Instead of partitioning the genome using only chromosome by chromosome, you could start partitioning the genome based on phenotypic annotation or based on epigenomic annotation, for example, DNA hypersensitive sites or promoter regions or UTRs or coding regions. And that's in fact at the basis of this stratified LD score regression that Alkis will present shortly. So anyway, it's really a pleasure to have here Alkis Price. He's a professor at the Harvard School of Public Health, and he's actually one of the developers of many of these models that have been really driving the field in many ways. So Alkis, take it away.
All right, well, good afternoon, everybody. I'm Alkis Price from the Harvard School of Public Health, and the title that I settled on for my brief presentation is Disease-Critical Cell Types and Cellular Processes Across the Human Body. And this does, as Manolis mentioned, make use of a tool called stratified LD score regression. When I'm teaching, I like to encourage people to jump in with any questions and comments you might have, both because it's more fun and because it may actually be more informative for everybody that way. So please don't be shy about jumping in. I know Manolis won't be. All right, so this is the outline of my presentation. And I'm going to start with the methods piece and not with the biological motivation piece. But if you would like to know what the biological motivation of why it would be, useful to humankind to learn about disease-critical cell types across the human body, you can find some of that biological motivation in this review paper. But on to the methods piece, and this piece is about stratified LD score regression. The most pertinent reference is Finucane 2015. And so I'm going to start with a definition of SNP heritability. Now, there are different definitions of heritability out there, all of which are important. This particular definition is a function of a particular set of SNPs. And so the underlying value of this quantity in the population is a function not only of the population that you're studying, obviously it's a function of which disease or trait you're studying, it's also a function of which set of SNPs you're studying. If you have a large set of SNPs, then that may have a larger SNP heritability associated to it than some small set of SNPs. And so with that in mind, we're going to define SNP heritability as a parameter that's defined in the entire population as the maximum value that you could attain in the entire population in terms of maximizing the R squared between your phenotype and any linear combination of the genotypes. So this is defined as a parameter in the entire population. I don't need to make any kind of special assumptions to make this definition. And then, of course, if I happen to have in hand data from some finite sample, then, if I'm lucky, I might have some good method that could provide a pretty good, unbiased, not too noisy estimate of this parameter, based on the finite amount of data that I have. And as Manolis noted a moment ago, the GCTA method of Yang et al. 2010 is the kind of, main paper or first big paper in this space of estimating SNP heritability from human genetic data. But I'm not going to go into the details of that right now. And so above and beyond estimating a single SNP heritability parameter across the entire genome, a wrinkle on that may be potentially very interesting is to define and estimate a SNP heritability parameter for different functional categories. And so when I talk about functional categories, we'll get into this, but you can think about coding versus non-coding SNPs, regulatory SNPs, SNPs that are conserved across mammals. There's different flavors of SNPs in the genome, and it's of interest to partition heritability across those different functional categories of SNPs. And so if we have multiple functional categories of SNPs, maybe we partitioned all SNPs into 5 or 10 or 20 different functional categories, then we can we can generalize this definition to variances explained by the set of SNPs in each respective category. And this can enable us to reach conclusions. Well, then we have to think about doing estimation in a finite sample, using data from a finite sample, which I'll get to momentarily. But this could enable us to draw conclusions like, coding SNPs are enriched for heritability, or, regulatory SNPs in a particular tissue are enriched for heritability of a particular disease, or SNPs that are conserved across mammals are enriched for heritability, or those types of conclusions. And again, there's a fundamental distinction, in virtually any problem in this field between, on the one hand, providing a definition of a parameter that is a valid definition considered for an entire population, and on the other hand, coming up with an estimation method, which given a finite amount of data in a finite sample, can endeavor to produce a good estimate, that's to say an unbiased estimate, hopefully with a low standard error of that population level parameter. All right, so in the early days of statistical genetics, everybody was analyzing individual level data, genotypes and phenotypes for each of thousands of individuals. But as we get into bigger data sets and disease consortia that are meta-analyzing summary association statistics from 40 different cohorts, as we move into this phase of the history of statistical genetics, there's an increased amount of interest in analyzing summary statistic data and devising methods to analyze summary statistic data. And this is in large part responsible, this 2012 Nature Genetics editorial kind of precipitated this, in which they didn't require it, but they requested that authors who publish in their journal publicly release their summary association statistics, where by summary association statistics, I'm talking about GWAS association z-scores for each SNP, genotyped or imputed SNP analyzed. And in some cases, it can be useful to know the sample size on which that z-score was computed, because there might be some SNPs which were poorly imputed and removed from a subset of the cohort, so the sample sizes may vary a little bit with the SNP, and it's useful to know that. And at the bottom of this slide, I've noted that in many of the applications, it's useful to have reference LD information, linkage disequilibrium information about correlations between SNPs, computed, for example, using some population reference panel such as 1,000 genomes. And so mostly today we'll be talking about methods that analyze summary statistic data, which certainly for most diseases you could think of, schizophrenia, rheumatoid arthritis, Crohn's disease, whatever disease you're interested in, chances are you're going to have the best opportunity to analyze very large sample sizes if you're willing to kind of willing to make do with summary statistic data rather than having complete access to individual level genotypes and phenotypes. All right, and so the method I'm going to talk about, stratified LD score regression, is a method, first providing a little intuition before we get into the math, is that we're going to regress chi-squared association statistics from a disease GWAS. This is a regression performed across SNPs, and we're going to regress the chi-squared association statistics with LD from different functional categories. And the idea here is that if I want to assess, for example, the hypothesis that there might be an excess of heritability in coding SNPs, I don't just want to look at the association statistics for coding SNPs and compare them to the association statistics for non-coding SNPs, because the association statistic, it's not just coming from the causal effects of a given SNP, it's also coming from the tagging effects of a given SNP, where you're tagging other causal SNPs. And that's why, the hypothesis might be something like SNPs that tag a lot of coding SNPs have higher chi-square statistics. That's kind of a more appropriate hypothesis to test than the hypothesis coding SNPs themselves have higher association statistics than other SNPs. And so this method relies on the very fundamental idea, due to various different Yang et al. Papers, that when you're running a GWAS, an average chi-squared greater than 1 does not imply confounding. So, back in the old days, a long time ago, people thought, well, if I'm running a GWAS, maybe using only a few thousand samples, nearly all the SNPs are probably null SNPs, which should, under the null, have an average chi-squared of 1. Or you can also compute the median chi-squared, which is a little bit more robust to outliers. That's called lambda genomic control, but I'm not going to get into that. At a small sample size, the average chi-squared should be pretty close to 1. In the old days, when your average chi-squared was substantially above 1, people would think, oh no, my average chi-squared is greater than 1. I must have some kind of problem with confounding due to, population stratification or cryptic relatedness or something like that. But people, realized over time, particularly in this Yang et al. 2011 paper, people realized over time that as we get to large sample sizes, we can expect our average chi-squared statistics to be greater than 1 due to true polygenic signals. And some of this math is from Yang et al. 2011, where we actually can write down as a function of the SNP heritability, well, both the SNP heritability and the amount of LD, the precise extent to which we expect the average chi-squared to be bigger than 1. And so this equation over here, this is an equation which literally specifies the precise amount by which you expect average chi-squared to be bigger than 1. And the more LD you have, this quantity here is the average LD score across SNPs, where an LD score is just a sum of R squareds that a SNP has with itself as well as other SNPs. The more LD you have, the higher the average chi-squared is going to be, because the chi-squared association statistic, the amount by which it exceeds 1 due to causal, due to true signal. Well, that true signal could be causal signal, where a SNP is biologically causal, but it could also be tagging signal, where a SNP is not biologically causal, but is correlated to some other SNP that's biologically causal, and thus has sort of true signal, an average chi-squared greater than 1. So this idea that goes back to Yang et al. 2011, that an average chi-squared and greater than 1 does not imply confounding, but is actually an expected consequence of a particular level of SNP heritability coupled with a large sample size. This is the idea that kind of lies at the heart of this method, stratified LD score regression. So we can start with LD score regression without functional categories, and it looked like from Manolis's outline he might have already gone over it, but we can still go over it. So here we're gonna regress for each SNP. This is a regression across SNPs. We're gonna regress for each SNP its chi-squared association statistic on that SNP's LD score. That is to say, it's sum of R-squareds with itself as well as other SNPs. And we expect that the intercept that we'll get from this regression should be equal to 1 if there's no confounding, or greater than 1 if there is confounding.
So in practice, do you need to worry about any SNP outside your LD block? How many LD blocks away?
In practice, we've done this, we've restricted the computation of the LD score to plus or minus 1 megabase, which which is generally sufficient. I mean, LD for common SNPs, typically only extends on average about, maybe 50 kb in European populations, going down to maybe 25 kb in African populations, which were not subject to the out of Africa bottleneck, which increased LD. That's on average. Obviously, you're going to have some LD spanning further than that, particularly as you go to low frequency SNPs. And so we elected, I think, in all of these papers to go to plus or minus 1 megabase.
Has anyone tried doing the full computation across all SNPs in the genome versus, say, just the 1 megabase neighborhood versus, say, a few LD blocks surrounding to see if there's any difference?
Yeah. So this gets complicated. I mean, number one, it might be very computationally intensive. Number one, it might be very computationally intensive. Number two, if you're looking at a reference LD sample, there really isn't going to be any across chromosome LD. Like it's sort of in some sense, especially if it's a very homogeneous LD reference panel, it's literally impossible for there to be a cross chromosome LD.
Carry on. Sorry for the distraction.
If you're so lucky to have individual level data and access to the within GWAS samples LD, there could be a cross chromosome LD due to chance, right? If I have a finite sample, and this happens particularly often for rare SNPs, if I have like a singleton on one chromosome might be correlated, to a singleton on another chromosome if it just happens by sheer chance that the reference allele is present in the same individuals for both of those SNPs that lie in different chromosomes. And the situation seems to be, one of the projects in our group led by postdoc Martin Zhang is kind of looking into these issues in the case of rare SNPs. And once you start to go to rare SNPs, then the issue that you just raised, Manolis, kind of becomes more important, and it becomes more appropriate to consider sort of chance, not population level, but sample level chance LD between chromosomes in the finite target sample. And there's theoretical reasons why, in principle, you're actually more interested in the sample LD in the target sample than in the LD in the entire population. But that's a subtlety that it's probably not worth going into given the finite amount of time we have, and it doesn't actually make a big difference. So the short answer is don't worry about it, especially if you're focused on common SNPs.
Beautiful.
All right. And so all of this stuff, might be most intuitively interpretable using a real data set. And so this is a real schizophrenia data set, from a landmark 2014 paper. And this is real data here. And on the x-axis, we have, now it's not the case that each point is one single SNP, because then it would be noisy and impossible to look at. But each SNP is like a collection of SNPs, an average of a set of SNPs with similar LD scores. So they're SNPs binned by LD scores, and these are LD score bins. And we can see on the right side of this plot, SNPs that have a ton of LD, like on average, they're an LD with like 250 different SNPs. Well, if you're an LD with 250 other SNPs, then you're potentially tagging a lot of SNPs. You're tagging a lot of SNPs that potentially might be causal. And so an expectation, you're going to have an average chi-squared because of all that signal that you're tagging from the other SNPs. Whereas if you have a low LD score, close to 1, which is the minimum LD score, because every SNP is going to be an LD with itself, as we look at these very low LD score SNPs, then it turns out when you run this regression, you see that they have average chi-scores very close to 1.0, which is basically reflecting the fact that if you're tagging close to 0 SNPs in total, then you are actually going to have null chi-squared association statistics, which is what you'd expect if there's no confounding. And so the fact that this intercept is close to 1, that once you kind of subtract out the effect of LD and you're not in LD with a ton of other SNPs that might be causal that you're tagging, then your average chi-squared is kind of close to 1. So average across the entire data set, the average chi-squared is 1.6, which is way bigger than 1. And the fact that this intercept, when you run this regression, comes out to 1 means that average, it's not due to confounding. It's predominantly due to polygenic signal in proportion to the number of SNPs that you're in LD with or that you tag. And the fact that this intercept is 1 implies, or close to 1, implies that it's not about confounding, it's about LD tagging of causal signal. So that's what it looks like if we don't have functional annotations. Well, that's the intercept tells you about confounding. Then the slope, if I may just briefly go back to the main equation here in this slide, the slope can be used to estimate this quantity, which gets you to the SNP heritability. And so the slope, it being equal to this quantity, can be used to estimate the SNP heritability. And we have to be a little bit careful when we talk about SNP heritability, which set of SNPs we're estimating the SNP heritability of. And it turns out in this case, we're estimating the SNP heritability of whichever set of SNPs is in our LD reference panel, such as 1,000 genomes. Now we can make this more complicated, add an interesting layer of complexity by saying, instead of just looking at, the total LD score of a SNP, we can look at the LD score of a SNP with respect to SNPs in a particular functional category. So like, for example, the LD score of a SNP with respect to coding SNPs. And if that's the sum of the R-scores of that SNP with all coding SNPs. And then now, instead of the LD score always being at least 1, if I take a non-coding SNP, it could, in principle, be not an LD with any coding SNPs, and it could, in principle, have an LD for coding SNPs of 0. It's possible. And so we compute these sort of LD scores for each functional category of interest to us in turn. And then instead of doing a sort of unilinear regression of chi-squared statistic on LD score, we can do a multilinear regression of chi-square statistics on the LD scores, on the respective LD scores of each of the functional categories that we're interested in. And it turns out that the respective slopes of that multilinear regression can tell you something about the SNP heritability that's causally due to SNPs in that particular functional category. So the causal SNP heritability of coding SNPs, the causal SNP heritability of regulatory SNPs or regulatory SNPs in a particular tissue, and so on and so forth. And there's a fundamental quantity enrichment, which is just like for a particular functional category, let's say coding SNPs, what's the percentage of SNP heritability explained by that functional category versus the percentage of SNPs that's in that functional category. So if it's 1% of SNPs, but it explains 10% of SNP heritability, then it's 10 times enriched for heritability.
How fine-grained can those subdivisions be? Because if you're within the same LD block and you have a bunch of different functional categories, every SNP is influencing every other SNP in that LD block, and yet you're partitioning them to different categories. Is there any?
I mean, the answer is it can be very fine-grained. I mean, you could have you can imagine a set of, I don't know, 100 SNPs that all have LD of 0.8 with each other. But if, in terms of if you're doing fine mapping at a single locus and you've got 100 SNPs that each have an LD of 0.8 with each other, it's probably a disaster for successfully fine mapping an individual locus. And that's why fine mapping an individual locus is a statistically incredibly hard problem. And most loci simply cannot be effectively fine mapped. But once you talk about integrating information about coding and regulatory SNPs and whatnot across an entire genome of data, then you can kind of start to tease that apart and actually get pretty precise estimates of what the heritability causally explained by coding SNPs or causally explained by regulatory SNPs and so on actually is.
Thank you.
All right, and so I've been a little bit, fast and loose about overlapping versus non-overlapping, but it actually works totally fine if the functional categories are overlapping. Like, maybe you have one functional annotation which has values 1 for coding SNPs and 0 for non-coding SNPs, and then you have a different functional annotation that has value 1 for SNPs that are conserved across mammals and 0 for SNPs that are not conserved across mammals. But of course, there's some level of overlap between coding SNPs and SNPs that are conserved across mammals. Those are overlapping functional categories. And it turns out it's totally fine if they're overlapping. We have to make the functional interpretation of the slope, if they're non-overlapping, we have some kind of simple, generalization of, n divided by m times SNP heritability. Once the functional categories are overlapping, it starts to get a little bit difficult to think about how we interpret this slope. But we can still interpret the slope if we do it carefully, and we can still, define enrichment in much the same way in terms of like, maybe coding SNPs might be 1% of all SNPs, but might explain 10% of heritability, and the same for a zillion other functional annotations that could be included in the same model. And we could even take it a step further and look at continuous functional annotations, which we did in a follow-up paper from 2017. And here again, I'm going to gloss over the details, other than to say that everything that you do with binary annotations, you can pretty much do all the same math with continuous valued functional annotations if you just introduce a few extra bells and whistles along the way. And I should mostly skip this slide, other than to say that we have an extra layer of complexity of introducing some regression weights to account for the fact that you have correlated data. If one SNP in the regression is correlated to another SNP in the regression, then that's redundant information, and we should correct for redundant information. And there's a different phenomenon, which is, it's actually a statistically correlated phenomenon, but it's a fundamentally different phenomenon, involving heteroscedasticity, in which we know that a SNP with higher LD scores has a higher mean and also variance on its chi-squared, and we should probably account for that as well, so that SNPs with high LD scores don't totally dominate the output of the regression. So those are some bells and whistles which we don't have to go over in detail. So in the original 2015 work, these are some of the functional categories that we looked at. Coding SNPs, SNPs that are conserved across mammals, various flavors of enhancers, histone marks, et cetera, And this is a set of simulations showing that, as we might expect, the method produces unbiased estimates in an idealized situation in which all of the causal categories, in this case, the causal category is DHS, which is a functional annotation, a DNase 1 hypersensitivity sites. It's a marker of open chromatin. And as long as the model knows about the causal categories, it should come as no surprise that LD score regression, regardless of whether we just tell it about these two categories, DHS and non-DHS, or whether we tell about it the full baseline model with all of these different annotations, that you could throw in 30 annotations in the model and do a multilinear regression on 30 annotation categories. Either way, it works pretty well, just as if you used individual level data REML methods to partition the heritability across the two categories. All those approaches produce basically unbiased estimates. But the tricky bit comes when you have causal categories that are not in the model. So, in this case, we have, you're going to use the same causal category, DHS, but we're going to not put that causal category in the LD score regression model. If we have like two categories, where we're looking at, or I guess we're telling the model about DHS and non-DHS, but then we're going to evaluate the enrichment of these other categories like coding or phantom 5 enhancer. And you can see that both REML individual level database methods and LD score regression methods sort of don't work very well when they don't know about these causal categories like flanking regions or coding regions or enhancer regions. And this isn't really a big surprise. If you lie to a model about what the truth is, and then the model then tries to do inference without knowing the full truth, it's probably going to produce a biased result. But the good news is, if we try to run LD score regression and we include all these, categories of the baseline model that we use when we run stratified LD score regression, and then we remove the causal category, but we retain all the other categories in the baseline model, then the good news is that stratified LD score regression still produces pretty close to unbiased estimates. And this is saying that the baseline model is rich enough, it's got enough different functional categories in there that it knows about, that even if there's a few of them that are missing, of course we should not believe that the baseline model literally captures literally every aspect of real life functional enrichment. It just includes a handful of 30 annotations that we know about. But the evidence seems to be that even if we remove a category or two, which is causal and which we try to do inference on, then the remaining 29 categories that are in the model seem to provide enough richness, enough modeling richness, that the model misspecification is minimal, and we're still able to produce at least pretty close to unbiased estimates. And that's, in comparison to if we either run LD score regression using just two categories like DHS and non-DHS, Or equivalently, if we run individual methods that analyze individual data just using DHS and non-DHS, then we run into problems. And the individual level data methods are not currently, have not been adapted or designed to run on like 30 overlapping or in some cases continuous valued functional annotation categories. So it's not really an option right now to say that we're going to run REML using a large number of overlapping categories or even continuous valued annotations. All right, there's a subtlety that I feel like I should skip over, where you can still violate model assumptions if you're not careful. If you look at like top cis-QTL in gene expression data, then that's an example of an annotation that doesn't satisfy the fundamental model assumption that any SNP that as a function of its annotations, SNP effect should be IID, because it turns out that SNPs that are in LD with top cis-QTL might be causal eQTL because top cis-QTL might be tagging. And so that the analysis of this annotation of top cis-QTL fundamentally violates the modeling assumption that as a function of the functional annotations that you're looking at, all SNPs are IID subject condition on their annotation values. And that modeling assumption turns out to be false if you look at an annotation called top cis-QTL, which can include a lot of tagging SNPs that just happen to be the top cis-QTL the locus. And you can get into biased estimates, both for SLDC as well as other methods when you do inference under those circumstances. All right, so I don't have a lot of time left. This is a natural time to switch gears from the methods piece to the real data piece, which to me is really the best way to kind of teach methods science, is to see what's going on when you look at real data. So now we're going to look at some of the applications of this method to some chromatin data and also some gene expression data. And maybe we might even get to LD dependent architectures. So these are the 17 traits that we analyzed in the initial work. These are all publicly available summary statistics in fairly large sample size. These days, 100,000 would count as fairly large and not extremely large. Extremely large would be like 500,000 or a million, but still fairly large sample size. And one of the initial findings is that coding SNPs actually are enriched for SNP heritability of diseases and complex traits, depending on which particular traits you're looking at. We see the largest enrichments for autoimmune diseases and height, and maybe slightly lower enrichments for schizophrenia and BMI. And likewise, different flavors of enhancer and enhancer-related annotations, various regulatory annotations are likewise enriched, maybe not as enriched as coding SNPs, but still have substantial excess enrichment for diseases and complex traits. And interestingly, SNPs that are conserved across 29 mammals were actually one of the most enriched functional categories, even more enriched for heritability than coding SNPs. And that's saying that SNPs that, in the sort of, mammalian phylogeny, where you have a SNP in humans in a part of the genome that has just not tolerated any mutations in the course of mammalian phylogeny, then that means that SNP is probably doing something important. And if it's doing something important, then it might have a pretty substantial impact on disease. And that's what this analysis is telling us. And then we looked at phantom 5 enhancers, which are a particular very specialized enhancer assay. And in the case of phantom 5 enhancers, we saw a very large and statistically significant enrichment for autoimmune diseases and nothing appreciable for the other traits.
Okay, should we also interpret these results as BMI has more to do with survival than height or schizophrenia or immune, or no, you wouldn't read into that?
That's a good question. I think well, we have to pay attention to the standard errors here. I think it's true. Well, so first of all, if we have a SNP that influences, let's take schizophrenia as example. Schizophrenia is a good example. If we have a SNP that increases your risk of schizophrenia, there's a point of view that says, well, if a SNP is going to increase your risk of schizophrenia, individuals with schizophrenia on average have fewer children. That's probably been true for tens or hundreds of thousands of years. Then, mutations which, increase your risk of schizophrenia probably cause you to have fewer children. But what the community actually believes is that there's something else that's going on, something called pleiotropic selection, where if you have a SNP that influences schizophrenia, it probably impacts the brain in all sorts of ways. And, it might even be a SNP that reduces your risk of schizophrenia, but it's still going to impact the brain in a profound way. And nearly all mutations, do have a bad impact on fitness. Now, of course, we know that there's a few mutations that have done great things that have caused us to be humans and not chimps over the past 5 million years, but nearly all the time, mutations do bad things. Even if it's a mutation that's going to reduce your risk of schizophrenia, it's probably doing a bad thing. And so there's some sort of unsigned coupling between SNPs that impact schizophrenia and SNPs that impact reproductive fitness. So there is some sort of coupling there, and maybe we might read into the sort of strength of that coupling being higher for some traits and lower for other traits. And maybe it might be higher for brain-related traits because brain-related traits, SNPs that impact brain-related traits, might be under strong negative selection, and maybe less so for autoimmune diseases, perhaps.
Yeah. And I also want to point out to the class, of course, that directionality doesn't matter. So it's not BMI per se for either super thin or super heavy. It's really that stuff that has to do with metabolism, period, right?
Yeah, and really, any SNP that impacts anything is probably, any SNP that impacts just about any disease or complex trait is probably impacting something else as well, which probably reduces fitness, or the derived allele is probably reducing fitness.
Great, carry on, thank you.
And why are these categories functionally enriched? It turns out in this paper on polygenicity, and I'm not going to get into the details of the method, the ultimate conclusion was that the reason why these functional categories are enriched for heritability it's actually not due to larger causal effect sizes. It's not like you have the same number of causal SNPs, but they have larger effect sizes. That's not true at all, because it turns out that common SNPs have some kind of soft upper bound on what the effect sizes can be due to the constraint of negative selection, where if a SNP does a lot of things, negative selection is not going to allow that SNP to increase to being common in the population. And so it's not so much about you have the same number of causal SNPs with larger effect sizes. Instead, it's you have more causal SNPs that each have this kind of like medium or small to medium effect size, causal effect size, due to this soft bound imposed by negative selection, conditional on becoming a common SNP. So it's really more about how many of the SNPs in the functional category do something, and it's not about like the SNPs that do something have huge effects.
Could you explain what low frequency means?
So low frequency here is low frequency is, so mostly we're talking about common SNPs. Low frequency SNPs are SNPs between 1 and 5% monoallele frequency. Right. And so both in terms of heritability enrichment and in terms of polygenicity enrichment, this means that low frequency SNPs are less polygenic. There tends to be less heritability per SNP, and they also tend to have sparser effects.
Isn't this a little counterintuitive?
Isn't what a little counterintuitive?
That they're at that end. You would expect that the low frequency are perhaps low frequency because they're being selected not to be high frequency.
Okay, so I should be a little bit more careful in how I explain this. The x-axis is heritability enrichment. For any given SNP, the amount of heritability explained by that SNP is proportional to its per allele effect size squared times p times 1 minus p, where p is the allele frequency. So on the one hand, we know that that rarer SNPs, such as low frequency SNPs, have larger per allele effect sizes. Each allele has a larger effect. But we also know that after you multiply by p times 1 minus p, they actually have smaller per SNP heritability. Yeah.
So the normalized one might actually be at the other end of the spectrum.
Yeah, if you were to look at per allele effect size, then rarer SNPs would on average have larger per allele effect sizes, or squared per allele effect sizes.
Thank you.
All right. And so now we're going to get into the part, we're finally at long last going to get into the part that corresponds to the title of the talk, having to do with inferring disease critical cell types. And so this is in the original 2015 paper looking at cell type specific regulatory annotations, looking at the ones that were most enriched for a particular disease. And this is obviously just a few of the most notable examples, but for schizophrenia, it's brain regulatory annotations that are most enriched. For height, it's connective bone regulatory annotations that are most enriched. And for rheumatoid arthritis, it's immune cell types that are most enriched. And this is, of course, very consistent with known biological knowledge. And this is a proof of concept, at least on this slide, that you can use cell type tissue or cell type specific regulatory annotations coupled with a disease heritability enrichment analysis using stratified score regression to infer disease critical tissues and cell types for diseases and complex traits. This is a larger set of traits on this slide, and some of these, including smoking and educational attainment, where it's not a huge surprise that the brain would be important, but these are results that had not previously been attained with genetic data due to a very limited number of genome-wide significant SNPs for these traits at the time that the Finucane 2015 study was conducted. I think it was 1 and 3 genome-wide significant loci respectively for these traits. And this is a demonstration, especially for these highly polygenic traits, that a genome-wide polygenic approach can, in some instances, yield greater biological insights than traditional approaches that focus their efforts on genome-wide significant SNPs, which may be very low in number for these highly polygenic traits. All right, so this is a follow-up paper of Finucane 2018, where we're using specifically expressed genes. So this is kind of a totally different approach. Now we're getting into gene expression data, and we're using specifically expressed genes. And so this is saying that genes that are specifically expressed in brain relative to other tissues are enriched for schizophrenia. Genes that are specifically expressed in connective tissues compared to other tissues are enriched heritability for height, and genes that are specifically expressed in blood cell types or immune cell types have enriched heritability for rheumatoid arthritis. So this is very much a complementary approach using a different type of data than the sort of cell type specific histone marks or chromatin marks that we were talking about a moment ago. And these results were in general broadly concordant with the results on chromatin data, but there are instances of data sets where you actually get better resolution in the gene expression data that's available as compared to the chromatin data that's available. So it's a complementary approach. I mentioned continuous functional annotation, so I am going to have to head out in a couple minutes, but there's a piece here on continuous functional annotation that has to do with LD-dependent architectures, where an LD-dependent architecture refers to the dependence of causal effect sizes on the level of LD of a SNP after conditioning on minor allele frequency. And we were very surprised to see that SNPs with lower levels of LD, even after you've adjusted for MAF, have larger causal effect sizes. And this is true for all 56 traits that we studied, from agent menarche to unibrow, where there's a fundamental effect where SNPs with lower LD have larger causal effect sizes. The story here is rich and complicated. It turns out to have something to do with negative selection. And there's reasons why negative selection kind of plays into these LD-related annotations and leads to this phenomenon that SNPs with lower levels of LD at a given minor allele frequency actually have larger causal effect sizes. So it looks like I've run out of time. I'm sure Manolis can share these slides with you, and you can see maybe on your own time the piece of this that has to do with analyzing single-cell RNA-seq data to learn at sort of more exquisite resolution some things about disease-critical cell types. Other than that, I'm going to have to go. I might have time for maybe another 1 or 2 very quick questions.
I want to really thank Alkis for a beautiful, beautiful presentation. So I don't think we should hold you any longer since you have actually a class starting at 2 o'clock. So thank you so much.
Okay, thanks for the invitation.
Thank you. Bye-bye. All right, Manny, I see that you're here as well. So it's a real pleasure to introduce Professor Manuel Rivas from Stanford University, a longtime friend and, in many ways collaborator, especially through his most recent student, Yosuke. So Manny, it's such a pleasure to talk to you. So he'll be telling us today about how do we combine electronic health records, genome-wide association studies, and genomics, basically taking what Alkis focused on, which is one trait at a time, and now asking, how does that apply when we look at the entire electronic health record of a very large cohort of individuals? Manny, take it away.
Thank you, Manolis. So it's a real pleasure to be here. At first, I'll describe some of the work that we're doing in terms of combining data across multiple phenotypes. So just give a brief overview. One of the data sets that, that we have been thoroughly studying is the UK Biobank data set. It's a population-based cohort of approximately 500,000 individuals with a mixture of electronic health records, web-based questionnaire data, genotyping, baseline biochemistry, and imaging data as well. So it's a rich resource with thousands of phenotypes and very rich data set on the genetic level as well. So the typical approach that we take for analyzing genetic data, so this study was led by Yosuke Tanigarawa. It's in Nature Communications, Components of Genetic Associations Across 2,000 Phenotypes in UK Biobank. So typically in a genetic study, what we do is to run linear regressions 1 million times over, say, 1 million genetic variants for a single phenotype, as some of the analysis that Alkis Price described. So typically what you find is polygenic characteristics where multiple genetic variants across the genome are associated to a given trait. Given that we had access to UK Biobank across thousands of phenotypes, we can also ask the question, for a given variant that we find to be associated with a phenotype, are there associations as well to other phenotypes among the thousands of phenotypes we have access to? And this is just one example of a genetic variant where we've analyzed the association for multiple phenotypes. So here, this genetic variant has association to many fat measures. Here on the x-axis is just the minus log 10 p-values of the linear regression model applied to those phenotypes. And on the right hand, we have a beta or effect size plot where we see the effect size across these multiple measurements that we have access to.
It also makes you question if they misinterpreted the question about being fed up.
That's right. That's right. Exactly. So sometimes you get some spurious associations as well, but other times it may be ascertainment issues. So one of the nice things about the characteristics of genetic association studies is that we have access to extreme polygenic traits, and we have pervasive pleiotropy, which sometimes limits the interpretability of genetic associations for. So here is just a diagram where we connect the genetic variants of the genome across thousands of traits, and you see many connections, mappings between the genetic variants and the traits. So one of the questions we asked is whether some of those connections can be described by a lower latent component that maybe can be best characterized by maybe hundreds of components. So some form of lower rank dimensional representation of the genetic association studies. And as Alka showed, for many of the traits that he's analyzed, he sees some shared characteristics. And that is definitely true of the UK Biobank data as well. So this approach we refer to as decomposition of genetic associations, where the idea is to disentangle many-to-many mappings into fewer components. So we refer to this approach as Degas after the painter Edgar Degas. And the idea is that for any given phenotype, as this pictorial diagram seems to show, for any given phenotype, you may be the genetic components may be shared across many other traits. So here's just an example of one phenotype with genetic components shared with the green component, the blue component, and the pink component, for example. So one of the things that we had access to was access to the genetic mapping of thousands of traits. So we can represent that data as a matrix W, where on the x-axis we have the variants, and on the y-axis we have the phenotypes. I think this data set was about 500,000 variants for 2,000 phenotypes. The matrix itself is composed of summary level data, so summary statistics, and that will be a theme that you'll hear more and more in this talk. And summary statistic is just of the flavor of a linear regression effect size estimate, along with a p-value or z-scores that corresponds to that p-value. So one of the approaches that we took was to just apply a truncated singular value decomposition approach to that matrix, and to represent that matrix in a lower rank component of about 100 components, 100 PCs. So each component is a product of orthogonal elements in these three matrices, U, S, and V of T. So you can see how a phenotype loads to each component, or how a matrix also, how a variant also loads to that component. So when we applied it to the data, the first two components, given that the UK Biobank is extremely rich in anthropometric measurements, you can see that the first two components of the data in terms of genetic associations is best characterized by the anthropometric phenotypes. So on the x-axis, we represent PC1, and the y-axis is PC2. And then you can see how each of the phenotypes loads onto these two components. For example, the projection of height sits squarely on in the middle between PC1 and PC2. Weight sits on PC1, and other measurements like hand grip strength, for example, are driven by a mixture of the two. Similarly, we could do this for the genetic variants, so to see how they load onto the two components. So here, for example, if we're looking at FTO, this genetic variant here, that's an intronic variant on FTO, you could see that it projects onto this component 4, which maps onto this component PC1, PC2, it's labeled 4, which maps onto the weight component. And similarly, other variants like MC4R and BDNF lie more on the weight component, whereas other genetic variants like SCN4A map a bit more on the height component of
Well, I have to
composition.
say that in a way, I feel that there's so much more richness because every one of these arrows should be plotted in a much higher dimensional space than 2.
That's right, exactly. Yeah, that's right. So to appreciate as you go down into just this characterizing a specific phenotype. So here, for example, if we're looking at body mass index, you can see the contribution of a mixture of components. So these are just the phenotypes that contribute to it. But here it's just describing PC2 and PC1. But these two components just make up about 60%, 65% of the variance, whereas a larger set of other components contribute to the remaining 35% of the variance. So here, just to characterize, PC2 corresponds more to the fat component of body mass index. If we're looking at the phenotypes that load onto that projection, and PC1 corresponds more to the fat-free mass component. So the composition and contribution components for a given trait can be quantified with some scores. And here is a way that you can give meaning to each of the components. So body mass index, not surprisingly, is made up of a fat component and a fat-free mass component. Now, this has consequences for how we think about genetic risk as well. So if you were to think about the genetic risk of BMI, the genetic risk of BMI would be contributed of a fat component and a fat-free component among the other components as well. So one way that we use this in order to study some specific genetic variants was looking at these protein truncating variants that are predicted to have strong functional consequences on gene function. One of the reasons that we're quite interested in these protein truncating variants is that they give pretty much direct inference as to what gene is involved, what variant is involved. In addition to that, we also have some PTVs that we identified to have strong effect sizes. Here, looking at the decomposition of genetic associations for PTVs, or these protein truncating variants, across the thousands of traits. Again, if we focus on PC1, and here for the PTVs, it's PC3 that makes up the second largest contribution to body mass index. We see a genetic variant here labeled as 2 that projects onto the component that corresponds to like fat-free mass and high cholesterol level. And that corresponds to this genetic variant or PTV in this gene called PD3B. So it has more of a high cholesterol, fat-free mass contribution to BMI. Whereas if we focus on 1, which corresponds to this axis here, this one corresponds to phenotype 5 and 6. And 5 corresponds to hip circumference and weight, so more of the fat component of body mass index. And one of the reasons why we're quite interested in this is because we want to identify genetic variants that may contribute to adipogenesis or lipolysis effects, rather than just having a fat-free effect on body mass index. And so one of the things that we did was to take some of these variants for functional follow-up. And GPR 151 does seem to have functional consequences on adipogenesis, which is quite nice. And here, if you were just to represent the association of that genetic variant across the set of phenotypes that we had access to, you can see that a large fraction of these are fat mass measurements, with some fat-free mass measurements as well associated with the genetic variant. So one of the things that we've done as well is to make available the genetic associations for 2,000 phenotypes on this website called Global Biobank Engine. And the idea is that it becomes a search portal for anybody to either search their favorite gene, their favorite variant or phenotype, and browse the set of associations that are available by combining the sets of rich phenotypes that's available in UK Biobank with the hundreds of thousands of genetic variants that are also available. It's a resource that's available online at biobankengine.stanford.edu and available to anybody in the world. And currently it supports a mixture of data, including exome sequencing data, array genotyping from UK Biobank, Million Veterans Program, Biobank Japan, and some meta-analysis as well, where the idea is to combine the summary statistics across the 3 biobanks in order to improve power for detecting genetic associations. Million Veterans Program is one of those projects based in the United States that has very rich combination of electronic health records with genetic data. So some of the ideas that come to bear here are also being applied in that setting as well, and in Biobank Japan. In fact, there's this one paper where we applied this singular value decomposition approach across summary statistics to the combination of UK Biobank and Biobank Japan summary statistics. So one area that I did want to share with you is also identifying these experiments of nature. So these mutations that are predicted to have strong effect on gene function that sometimes have the effect of lowering risk for disease. And one of the reasons why we're quite excited about that space is because then it does lead to some therapeutic hypotheses that we may want to follow up by mimicking the effects of those mutations. And it can guide the selection of targets for drug discovery as well. And I'll just give you a few examples of those where we've succeeded in identifying some strong effect mutations. So just to give you a snapshot of the data sets that we're looking at data sets that are quite massive in scale, from millions of samples to tens of thousands of measurements. So how do you then identify and hone in, and tons of millions of gender variants? So how do you hone in into specific genetic hypotheses that you want to follow up later? One way to do that is by going from these big data sets to small data sets where you have summary statistics, and diving deep into genetic variants that are predicted to have strong functional effects and a strong effect size as well. In the UK Biobank, we've identified loss of function truncating variants that are predicted to break the protein coding sequence of genes. And in the study that we ran in 2018, we tried to identify individuals that carry these human knockout mutations, individuals that were likely to have a broken copy of a gene. And in fact, instead of conferring risk to disease, these individuals seem to have conferred protection against disease. One example of that is a protein truncating variant that protects against asthma. By looking at multiple measurements, including blood biomarkers, we were able to identify that these mutations ended up having strong lowering effects on eosinophil counts, and also having about a 2-fold to 3-fold decreased risk against asthma in this gene called IL-33. Similarly, in a gene called IFIH1, we identified strong effect mutations that lowered the risk for type 1 diabetes, lowered the risk for hypothyroidism, psoriasis, but also seems to increase risk for other diseases like ulcerative colitis. Nonetheless, paving the way to a pretty strong therapeutic hypothesis because the effect size were about 2-fold conferred protection against type 1 diabetes and about 1.5-fold effects for psoriasis and hypothyroidism. So a theme from this is basically the idea that we learn from these experiments of nature across populations. And one of the areas that we've put a lot of effort into is making these resources available so that we can improve inference by combining summary statistics across multiple biobanks. So first, I gave you a flavor as to what you do if you have access to data from a single biobank with thousands of phenotypes. Now, what do you do in the setting of having access to multiple biobanks? And one powerful way to improve your inference of the connection between genetic variants and phenotypes is to combine at the level of data, of the summary level data. And that's what we've done with other biobanks like FinGen and Biobank Japan and Million Veterans Program. And it just give you a highlight as to how you can identify specific genetic variants with strong effects on gene function, but also have strong effects on phenotypes. One of the things that we've done is to combine these phenotypes in order to visualize the relationship between the genetics of human traits and diseases. So we've estimated genetic parameters like correlation of genetic effects and heritability or polygenicity of disease, which you've heard a bit more about from August Price talk, where the idea is to characterize the extent to which the contribution of genetic variants contributes to the phenotypic variance for any given trait and how that relates in terms of the covariance of specific pairs of traits as well. So lastly, one of the things that I did want to focus in on is the concept of polygenic risk models. So the first part basically emphasizes how to use genotype data or genetic data with phenotype data to improve inference. So identifying genetic variants that you may want to either functionally follow up or maybe provide a therapeutic hypothesis, or maybe describe, this set of genetic variants are linked to these hundreds of phenotypes together in some lower rank representation component. But one of the other activities that we're quite excited about is risk prediction. So how can you use genetic data to improve your ability to predict disease risk? So one theme from the application of statistics to human genetic data is that typically the way we think about the relationship between a genetic variant and a phenotype is in terms of a univariate model. So you apply a linear regression or logistic regression, one variant, one phenotype at a time, and you apply that 1 million times over and over again. And then if you had access to thousands of phenotypes, you would apply 1 million regressions 1,000 times independently of each other. Now, this does provide the benefit of giving direct interpretation. It does provide some convenient computation, and it's fine with small sample sizes. However, for prediction, it has pretty weak properties because many genetic variants, as we've learned, come correlated with each other. So it's quite hard to distinguish which is the one that should be involved in your model, because if the task is for prediction, you'd be double counting it over and over again because there are just many variants that are come correlated with each other. That problem is referred to as linkage disequilibrium, which basically refers to correlation among the genetic variants. So one other approach is to take this multivariate model. The idea is basically that you fit one very large regression model where Y is your response, and then X, the predictor, becomes a very large model of millions of variables. The challenge is that it's less interpretable. It does introduce some high computational cost. You do need more data. However, since you've set it up as a predictive problem, it does lead to better predictive performance. So in terms of terminology, in the UK Biobank, we refer to these problems as large-scale problems because there are hundreds of thousands of samples, so N is very large. We refer to the problems as high-dimensional because there are hundreds of thousands of variables as well, given that we have hundreds of thousands of genetic variants to fit into one model. The package that we've developed in order to fit a multivariate regression model, to basically fit a response to all million genetic variants at a time, is referred to as SNPNet package. And the idea is that here we implement LASSO, the LASSO algorithm, which is a penalized regression framework that was developed by Rob Tibshirani and Trevor Hasty. So these are the co-authors of the paper, led by Young-Yang Piang, who just graduated from Stanford, who led this work. Here on the x-axis is just a representation as to how this model fit works. On the x-axis, since this is penalized regression, you have some lambda index that tells you how penalized the model should be. So as you go from 0 to 80, the less penalization occurs. And as you see on x-axis 2, the more variables that are included in your model. On the y-axis, you have r squared, which is the variance explained for the height that we're trying to predict. Here, the trait that we're representing is standing height. On the red and black curve will represent LASSO. The black curve corresponds to the fit of the model on the training data, and red corresponds to the fit of the model on the validation data. And as we'd expect, the model is supposed to saturate at a certain moment in time with the validation data. And that is when we decide to tell the model to stop adding variables. So here for standing height, we've included 47,000 variables into the model that gives a prediction or performance of about R² of about 0.70 for standing height. The green and the blue curve represent another model, which is referred to as relaxed lasso. The idea is that you use lasso for variable selection, but that you don't penalize the regression coefficients. So one of the observations here, at least, is that penalizing the regression coefficients does allow you to get better improvement in predictive performance relative to an unpenalized form. So these methods are implemented for anybody who's interested. We also have implemented elastic net regression, which has an L1 and L2 penalty. The models are a bit denser, but it has comparable predictive performance to lasso as well. So we've applied these polygenic risk score models for 35 biomarkers. This work was led by Yosuke Tanigawa and NASA's Armstrong. The idea was that we end up predicting predictive models for these biomarkers that are cardiovascular biomarkers, renal biomarkers, liver biomarkers. First, we created a 70% training data set, 10% validation to help us choose when to stop fitting the model, and then a 20% test split that allowed us to then say, this is the performance of the models when you've trained it across the UK Biobank. And similarly, we've applied it to additional test set populations, including non-British Europeans, South Asian, East Asian, and African populations. And one of the lessons that we've taken from this is that there is some limitation in transferring or porting over these polygenic risk scores over to non-European populations. So here's just an example of the variance explained in the white British component of the UK Biobank. Is about 50% for this biomarker called lipoprotein A. For LDL, it's about 22% for the white British European subset of UK Biobank. In the non-British European, the performance is quite comparable as well. So about 40% and about 20% for LDL. But now as you move to South Asian, East Asian, and African population, you see the performance of these models drop dramatically. And there are many reasons for it. Amongst them are the differences in correlation structure across the different populations. So transferring over these predictive models from one population that uses proxies of causal variance for prediction may not work as well in other populations because those proxies end up breaking down in terms of the relationship of correlation structure amongst the genetic variants. And in addition to that, you also have different sets of genetic variants that may explain the heritability of the disease or the phenotype, here being the biomarker. And just to give you an overall high-level summary as to how this looks, the notable exception is total bilirubin and direct bilirubin, where in the African population still has quite comparable performance to the white British or European subset in UK Biobank. But you can see that there's a dramatic drop in performance, about 80% drop in performance as we move to the African population. So this calls for the need for these population-based cohorts that combine EHRs, genetic data in non-European populations. And I think that's one of the more exciting areas of research in human genetics over the next years, is really starting to bring some of that data together.
Manuel,
yes.
I mean, this is fascinating, and I couldn't agree more about the importance of tapping into sort of the broader human genetic variation rather than this tiny little subset that is captured by the European samples that most of GWAS has focused on. I'm curious about the sources of that variation, because I think these can sort of have big effects in guiding a lot of the study designs. The one reason for the discrepancy could simply be that the genetic variants that are responsible for a lot of the European variation are simply not even found in Africa. These are variants that may have arisen after the out of Africa event, or at least, sort of greatly increased in frequency in the out of Africa event by sort of capturing a subset of the variation and bringing it to higher frequency. That's one option. The variant is not even there. Another option is that the variant is there, but doesn't have the same effect in the context of different environments. And when I'm talking about environments, I also mean the cellular environment of all of the other genetic variants that the variant is acting within. So, and for that, at mixed populations, like, African Americans, for example, can give you a sense of how much is the rest of the genome contributing to the differential effect of that one variant. I'm curious if you've looked into these, or if you have some insights to share through your group or other groups as to whether it's absence of variants or actually different effects of these variants. I mean, in addition, of course, to the LD component that you mentioned.
Absolutely. I think the absence of variants is a major contributor in the context of inflammatory bowel disease. When you move from European to East Asian population, for example, you do see the absence of some of the variants in the East Asian populations that are major contributors in European populations. In terms of effect size distribution or homogeneity of effect sizes, we do see when the variant is present across multiple populations, the effect sizes are quite homogeneous. There are examples of heterogeneity though. And I agree with you that, one way to get at that problem is to look at admixed populations. But the absence of the variants is a huge contributor. Yeah. And that's one example where, looking at, for example, lipoprotein A is a very simple example. The reason is that lipoprotein A has maybe tens of genetic variants that contribute to the variants. They're all in lipoprotein A region, right? They're a mixture of non-coding variants, coding variants, and even structural variants. The reasons why it performs so poorly in the African population is that the proxies for those genetic variants that are captured in the European populations don't exist.
Yeah. Thank you.
Okay, great. And with that, I'd like to end by just thanking everybody in the lab that has contributed to this, and largely Yosuke Tanigawa, who I'm very happy will be joining Manuel's lab.
Awesome. Manuel, thank you so much for a beautiful lecture. We have time for one or two quick questions, but I mostly want to thank both Manuel and Alekis for two awesome presentations. So Manuel, I mean, we are so fortunate to be able to have people of your caliber join us on, just for even 30 minutes. I mean, the proximity that we've had despite the distancing of the proximal people is one of the perks, I have to say, of this horrible situation that we're in. One of the questions that I always ask our guests is, will you be able and willing to sort of supervise students who are interested in doing research projects in these topics, especially with UK Biobank, polygenic scores, and pleiotropic effects?
Absolutely, would love to.
Awesome, very cool. All right, well, thank you very much, Manuel.
All right, thank you.
I think we have just thank you in the chat. All right, awesome. Thanks everyone, and then see you next week.
Bye-bye.
Well, thank you. The TAs can around.